It's a privilege and a pleasure to be here, and I hope to instill more hope, just as you heard about early this morning, by giving a general discussion of the pace of research for rare diseases. So I want to tell you a little bit about me first. This picture is the town where I grew up. That photo is from 1948, and the person on the left side of the picture is myself when I was 3. And what my mother complained about was that for the next 18 years, the most frequent word out of my mouth was why. Why does that happen? Why is the sky blue? Why? And that curiosity has continued for many years and is why I got into science. So I'm a pediatric cardiologist. I take care of kids with heart disease. But as Sue mentioned, and thank you, Sue, for getting me into this role, I was an educator as the chair of the Department of Pediatrics for physicians and scientists, but I also did a lot of research in rare diseases, not NBIA disorders, but other rare diseases. And I loved all of those roles. So what is the connection? First, as I said, I have an interest in rare diseases, and that's through mitochondria. And as most of you well know, NBIA disorders, many of them are focused in mitochondria. So this is a diagram of a cell, because we're going to be talking about that a little bit for the next 45 minutes. And it's complicated. There are a lot of different parts called organelles to the cells, of which the mitochondria are just one. And what do mitochondria do? They make energy. But they're also very much about things like cell death. And they're very critical to something called autophagy, which is a key component of some of these disorders. And they have lots of iron-sulfur clusters. And they have reactive oxygen species, all of which may be part of the pathogenesis of various NBIA disorders. And so my expertise is in mitochondria, and particularly in fatty acid oxidation disorders. So fatty acids, everybody says fat's bad for you. And too much is, as I personally can testify. But a little fatty acid is essential because your heart makes 70% of its energy from fat. And there are rare diseases that affect that pathway that cause severe symptoms and death in children, and that's what I've studied for the last 40 years. Iron-sulfur clusters, which are shown in this slide in mitochondria, are a critical part of making that energy, and they're shown down there in red for iron and yellow for sulfur. And so they're absolutely critical for being able to normally make energy. And they're involved in many processes within mitochondria that are crucial for all of its functions. And so when they're disrupted, as they are in some diseases, there's a major issue and phenotype. I want to bring this up because it's so important to think about. The work that we did on fatty acid oxidation disorders, along with hundreds of other investigators, resulted in newborn screening for those disorders. And those disorders are now screened for in every state in the United States, on every child born in the United States and many other countries. And newborn screening for those disorders has resulted in the ability to detect those disorders at birth. And many of the patients are then evaluated and treated immediately after birth. Shown in this slide are Robert Guthrie on the lower side, who was the first person to develop newborn screening for a disorder called phenylketonuria, and Helen Berry, who is from Cincinnati, who was very much involved in sickle cell newborn screening. Can that be done for NBIA disorders? I don't know, but it's something to think about because NBIA disorders, obviously, if diagnosed early, could potentially be treated early, and some of the complications might be prevented. Sue Lapola mentioned that I was at Cincinnati Children's, and I show this slide because what we did there corresponds exactly to what this organization is about: discovery and cure. So many years ago, Albert Sabin at Cincinnati Children developed the oral polio vaccine. How many of you got the oral polio vaccine? How many of you got polio? None. And there are very few patients in the world who get polio because Dr. Sabin invented that vaccine. Now, there are others who contributed in a major way, but he did his major work in Cincinnati. The first use of a heart-lung machine in a human being successfully was in Cincinnati Children's in 1952. And surfactant was cloned in 1988. That's the compound that's critical for survival of newborn premature infants. And so discovery is what we do at Cincinnati Children's. It's what many pediatric departments do, and the science is crucial to being able to cure and treat rare disorders. So what are rare disorders? And I want to spend a few moments talking about this because many of you say, why me? And you know, we have the rarest disorder in the world, and that's partially true, but there are many rare diseases. The definition is that they affect 1 in less than 2,000 individuals. There are over 6,000 rare diseases that have been documented. 80% of them are caused by genetic issue, just like NBIA disorders. And there are nearly 4,000 genes, probably by now, that have mutations that cause these disorders. And 10%, 8% of the American population is affected by a rare disorder. 8%. So you're not alone at all. And worldwide, there are 350 million people at least who have rare disorders. And half of them are children. And that's just illustrated in this diagram. But to emphasize that 350 million people worldwide have one of these disorders. And that's why learning how to treat them is so crucial. I'm not going to read this slide because it says what you already unfortunately know in great depth, but I show it to help you realize that many people now understand what you're going through. So rare disease patients and their families, caregivers, and health care providers face unique challenges. As you heard this morning, rare conditions are often poorly diagnosed and not well understood. The average delay in diagnosis for a rare disorder means that patients see 7.5 doctors. I'm a doctor. I wouldn't want to go through 7.5 of us, believe me, to get to where you need to be. And patients and families bear a huge burden from the effects of their disease. So this is a statement from a report about rare disorders in the United States that's made annually and emphasizes the importance of these disorders and finding what to do about them. And in fact, there's a national organization of rare diseases. How many of you know about this or have been to one of their conferences? So it's a pretty substantial organization, but it really is crucial because of what it's been able to accomplish. So why is this organization so important? In 1962, a law was passed that required the proof of effectiveness and safety of drugs before approval. That makes sense to all of us. The issue is how it's done and how difficult it is to do. So fewer than 40 drugs prior to 1980 were approved for rare disorders. Why? You know why. Pharmaceutical industry didn't have the resources to develop those drugs. They were hard to get past, and the margins of profitability from them would be small, they thought. And so the Orphan Drug Act was passed in 1983 to provide incentives for drug manufacturers to provide treatment for rare diseases. And in that drug act, 7-year market exclusivity was developed. Tax credit for drug development would reduce the cost of development of those drugs. And the Orphan Product Grant Program provides substantial subsidies for clinical trials, making it cheaper to get the drug to market. And some of them are exempt from the usual FDA fees, which can be substantial. So what was the impact of that? If you look at the third bullet point, by 2018, there were 503 approved orphan therapies for 731 different diseases or indications. So there was a very rapid increase in the development of drugs for rare or orphan diseases. And so that act has made a huge difference in what pharmaceutical companies and investigators are doing. Now, the arrow on this slide shows that 60% of these drugs in 2017 were for cancer. You don't think of cancer as a rare disease, but in fact, it's 1,000 rare diseases with different mutations that cause the same phenotype, let's say lung cancer. And so developing specific drugs for those mutations, to treat those mutations in patients with cancer, has become a very high priority, so-called precision medicine. And in fact, that's what is really one of the things that's really pushing this. If you look down a slide a little bit, the number of drugs being developed for patients with metabolic defects is substantial, but a lot lower. So the Orphan Drug Act has worked, but not always for extremely rare metabolic diseases. And the diagram on the bottom of this slide just shows how quickly the number of drugs, shown in red, has increased over the last years. And in 2017, there were treatments for rare disorders, including SMA that you heard about this morning, ALS, Lou Gehrig's disease, and hemophilia. And these advances are becoming more and more clear as to their efficacy in treating patients. So the one first main point I want to make is rapid progress is being made. It's not the same as it was 15 years ago. And that should give us all hope that drugs can be developed to treat NBIA disorders. Now I want to switch gears and talk about the steps in rare disease research. So the phenotype is what you know your family member has. What are the clinical signs and symptoms of the disorder? And usually that starts with somebody who describes either those symptoms and writes a medical report, and that is the initial disorder description. It's almost always patients who have the most severe form of the disorder, and the focus is usually on one organ system, in this case often the brain, because that's the main symptom, or because that's the specialty expertise of the physician who describes it. But in fact, as you also well know, most of these disorders impact multiple organ systems. So what's the next step? Finding the cause. And as you heard Patty say yesterday, or this morning, there were no genetic causes for these disorders back when this conference started, but that has rapidly changed. And I want to talk a little bit about why. So one of the keys is recognizing that these disorders obviously occur in families and what the pattern of inheritance is. Does it go from parent to child, dominant, is it recessive, et cetera. But the key to being able to make the diagnosis was the Human Genome Project. And now that Human Genome Project has allowed whole genome or whole exome sequencing of patients and families with rare disorders, and often, not always, often establishes the diagnosis. So what was the Human Genome Project? It started back in the late 1990s. It actually required multiple years to get Congress to think about, and the NIH to think about improving the human genome. And I'm old enough to know 3 or 4 of the individuals who were involved in those discussions and had to argue vehemently to proceed with sequencing the human genome. The first draft of a sequence was published in 2001 in Science. And a couple of the other landmarks were that it was determined in 2003 that, in fact, most of the human genome had been sequenced, and it was actually complete in 2004. It cost $437 million. The estimate of the cost of the Human Genome Project before it was started was $3 billion. In fact, $437 million is among the biggest bargains in history. And it has allowed us to know every single piece of the human genome, and we know that there are about 25,000 genes. Now, I do want to make one point here. That is that we have fewer genes than worms. And the difference among us is about 1% of all of the nucleotides in the human genome. And we're only 1.5% different from chimpanzees. So how different are we really? And I use this example to emphasize we're all in this together. And we need to know everybody's genome so that we can figure out why they get the problems they get. Once it's established, as it was back in the early 2000s for NBIA disorders, do all patients have mutations in the same gene? The answer was no, but for many rare diseases, the answer is yes. If there are multiple different genes that cause the same phenotype, how might they be related? That is, are they part of a pathway? And for an individual gene, what is the function of the protein product? And how do the mutations impact that function? So I tried to explain this to my wife this morning. She said, well, how do you get a protein? And I said, well, you start with the gene. And that gene, in a process called transcription, is made into something called a messenger RNA. And that happens inside the nucleus of the cell. The messenger RNA then goes outside the nucleus and is translated into a protein. A protein has amino acids. And that protein is what does the work. So you all know what gene and therefore what protein patients in your families have that's mutated. But it's the protein which is the end product of function. NBIAs do share some features, and there are a couple that are in the same pathway, but they're actually, as you know, very different, and they involve different parts of the cell. The arrow on this slide shows a mitochondrion, which is where, for example, PCAN lies, and a couple of the other disorders, but some of the other NBIA disorders damage proteins that work elsewhere in the cell. So there's not an obvious shared pathway. But there is the one, which is the CoA synthesis pathway, and you've heard a bit about that from vitamin B5, pantothenate, and the pantothenate kinases from PCAN are the block in the first step in this pathway. And so the question becomes, how can you overcome this defect? Can you bypass the block? And that's what some of the drugs are about. Can you induce the sister enzymes, PK1 and PK3, and have them function in the right place to cure the disorder? And the answer is probably. And can we induce the cell to make what it needs, the coenzyme A, another way? All of those approaches can work, have worked in other disorders, and may work in some of these disorders. But the process has to be corrected in the right organs, the brain in this case, the right cells, the neurons, and the right organelle, the mitochondria. And that's where it gets really hard. So a little bit more, genotype is whatever the mutation is, and there are many different varieties of those, some of which result in no protein being made, and some of which result in some protein which is partially active, which is why you get a whole span of severity for most genetic disorders. And the therapeutic approach will depend upon what the genotype is, what part of the process needs to be corrected. And then what happens after the initial delineation of the gene is that the phenotype is expanded. So mutations that result in no protein have a severe phenotype, and those usually need some sort of replacement. Missense mutations, which only change one little part of the protein, may have a mild phenotype. They may have some activity, and if you can increase that activity, it may treat the condition. Now, this slide is from Susie Jakowski's article from a few years ago, which talks about PK2 mutations. Patients have all, as you know, have all varieties of mutations. They have different impacts on function. Some are null and should require replacement, and others allow different therapeutic strategies, which are actually happening. But the only way to know how to approach it is to know what the mutation does. And what Dr. Jakowski did was to define the structure of PK3 in this case in very great detail and show where the mutations are and how they impact the function. And that was a key step in thinking about therapy. I want to talk a little bit about models as a third component of this talk because they're so crucial to developing therapies. And the mouse shown in the upper right is is a PK mouse, and you can see that the mouse affected has slow growth. Why do we do this? To understand what the gene does, just as importantly to understand how the animal adapts to the gene loss, look at all of the tissues in the animal to see which ones are affected, even if we can't test them all, characterize the course of a disease, so a mouse goes from to puberty within a few weeks, which allows very rapid delineation of what the course is, discover possible therapies, and then study the outcomes. Permanent cell lines are often used, derived from tissues like liver, kidney, and skin. They're readily available and they're well characterized, but they're all transformed. That is, they're like cancers. Many are not from humans. They're not particularly metabolically active, and they're not an organ. They don't have the multiple different cell types that, for example, the brain has. So one of the keys is something called induced pluripotent stem cells. How many of you have read about these? A few. These are really important. So if you follow this diagram, skin cells are taken from a person. They are treated in a way that makes them into stem cells, and those stem cells can form every single kind of different cell that we have by treating them in various ways. Not only that, if those cells are derived from a patient with a genetic disorder, that patient's abnormal gene can be edited and corrected in those cells. And then they can be differentiated into healthy cells and potentially be used for replacement therapy in a patient. Just as importantly, on the other half of this slide, is that we can use those cells to understand the process by which an organ forms and to target therapies. So their advantages are that can be derived from both animals and humans. They can become any cell type. They can be derived from patients with specific genetic disorders easily. And they can become organoids, which are organs that have cells that interact. They are not easy to work with, however. And here's a picture of one of these from investigator Jim Wells at Cincinnati. That's an intestine made from one cell. Every different color on that intestine is a different cell type. And it's a functioning intestine. It can take up food. It can secrete hormones. So it really lets us study what goes wrong, for example, in ulcers when it's infected with H. pylori, or in patients' diseases. and the mutation can be corrected, and it can grow artificial organs. And even though this is not exactly a brain, it's brain-like, derived from human iPS cells. And drugs can be discovered. So these little iPS cells can be put into hundreds of different wells on a plate in the middle, and thousands of drugs can be passed over those cells. And if there's an assay for correction, as shown by the analysis slide, an impact on the disease can be discovered. And that candidate molecule can then be used in much more complex ways to study the disease process. And knockouts are when we take an animal, usually a mouse, delete the normal gene, study the consequences, but it can be deleted in any specific cell, one cell type in one part of the body, and it can be induced at different times after the animal is developing so that we can analyze the disease process. And it allows tremendously in-depth studies. So that's why these animals are so important, and they can then be used to test therapies. I want to spend the last few minutes talking about gene therapy. So on the upper right side of this slide is a virus, adeno-associated virus. Now, perhaps the most commonly used vector for gene therapy. What's a vector? A vector is a piece of DNA in which you put another piece of DNA, and you make a virus, and the virus binds to the cells, let's say in the brain, and delivers that piece of DNA into those cells. And for example, and I will give you 3 examples, that can be the normal gene, not the mutant gene. The vectors can either go into the patient's DNA, become part of their DNA forever, or they can be not in the DNA and be diluted out when the cells divide. So what are the issues? There have been issues for the 20 years that gene therapy has been tried. Sometimes the patient already has immunity to the vector and then can't be used. And that's what caused early deaths in gene therapy. Not enough cells can take up the vector so that it won't have the desired effect. Sometimes it's very difficult to deliver the vector to the correct cells and only those cells, in the case of the brain because of the blood-brain barrier. And there are complications. So some patients have developed cancer. But there are successes, and I want to show 3 of them. First is severe combined immunodeficiency, bubble boy disease. That patient many years ago is shown on the upper left-hand side. On the upper right side and that patient was put in a bubble because if he was exposed to humans with infections, he would die. On the upper right is a patient treated by gene therapy for this disorder, obviously interacting with his mother and doing well a couple of years, but now many years after therapy. So the way this was treated, and it's a disease of white blood cells, so those white blood cells are taken out of the patient. A vector with a virus is put into those cells to correct the defect. It's put back into the patient, and in 20 patients in which that was done, 85% of them were disease-free for up to 15 years. This is an always fatal disease. 15 years, disease-free. There's another variety that's also been treated, 40 patients, 100% survival, disease-free survival, and 75%. And by the way, all of those patients are now all newborns are now screened for that disease. Sickle cell disease, and the 2 arrows here show the severity of this. 100,000 kids with sickle cell disease in the United States, 2,000 born every year. Their average survival is to age 40. But in Africa, there are 15 million people with sickle cell disease. The sickle cells are those pink things in the bottom right. that don't look like donuts. The red blood cells are supposed to look like donuts. So Poonam Malik in Cincinnati Children's has developed a gene therapy for this disorder. 2 patients have been treated in a prospective trial. And the difference is shown where the red arrows are. One of them went from being hospitalized 13 times in 1.5 years to none. And the second patient went from having severe pain crises 20 times in 18 months to 3. Both were weaned off opioid treatment for their pain. It works. It's not perfect, but it works. SMA you heard about this morning, except that what you heard about was a mild variety, not the severe variety. The infantile SMA type 1 develops weakness within the first 6 months, and without severe intervention like ventilators, and all of those patients die by the age of 2. The antisense oligonucleotide is a drug you heard about this morning, and it corrects the defect in one way. But gene therapy has now been done for SMA with an adeno-associated virus. 1 dose given in the blood, 1 dose, gets into the brain. Study was started in 2014. 15 patients were treated, all are alive, and they are much, much improved, some of them walking and talking. And that gene therapy was approved last week by the FDA. It is 5 years from the start of the trial and 2 years after reporting the results. That's fast. Why does that matter to you? Your kids don't have SMA, right? Why do I care? Because it's a precedent for the FDA. They can say this vector put into patients is safe, gets into the brain, and works. Is that what you want to hear? So gene therapy is not a cure-all and be-all because of the way it's done. It's not really the whole gene. It's not regulated the way the normal gene is. It may end up in cells where it normally would not be. It certainly, usually will not fully correct, and the mutant gene is still around, and there definitely will be complications and side effects. Having said that, for the first time, I believe that gene therapy will work for many, many rare diseases. So in conclusion, rare diseases affect millions of patients worldwide. Very rapid progress has been made in the approval of new therapies for rare diseases in the last 10 years. Animal models, especially iPSC cells, induced pluripotent stem cells, are allowing us to develop, evaluate new therapies, and gene therapy is working, although I caution it's very early and there are many uncertainties. Thank you for your attention.
Are you willing to take questions?
I'm sorry?
Are okay to ask questions?
Yeah.
All right. Does anybody have any questions for Dr. Strauss?
Hi, my son, in addition to having BPAN, has severe hemophilia as well, and so I've seen the rapid advancement in treatment in that in the last 5 to 10 years even, but realize it's also been around for hundreds of years. So could you speak a little bit to the timeline where a lot of these NBIA disorders are, you know, just in family history, study stage, and what that timeline typically looks like?
Yes and no. It depends on the type of therapy that one wishes to or thinks can be successful. Because so much of developing therapies depends upon understanding how the disease process occurs and what steps might be impacted, let's say, by a small molecule drug. So that takes time and investigators, and often ends up as a dead end. One of the reasons I'm enthusiastic about gene therapy is once you know what the genetic disorder is and where the abnormal protein is and where you need to put the normal protein, you can get going on that without knowing everything about the disease process. And as I tried to emphasize, because of the precedence in other disorders, the timeline can be, I'm not saying it will be, but can be dramatically shortened.
Thank you. Any other questions?
Thank you all.
Thank you, Dr. Strauss.